Nitrogen is an essential nutrient that plants require in large amounts for growth and development. Nitrogen is used to synthesize amino acids, proteins, and many other important metabolites, including vitamins. In industrial countries, high nitrogen fertilization guarantees maximum crop yields. However, this intensive level of fertilizer production and application is not sustainable due to the high energy costs, as well as negative effects such as increased greenhouse gas emissions and the leaching of the nitrogen fertilizer into aquatic systems, causing toxic algal blooms and dead zones. In developing countries, on the other hand, access to this nitrogen fertilizer is limited, resulting in reduced crop supply and ultimately food supply. One solution to both of these issues could be the production of crops that more efficiently use nitrogen and therefore require less fertilizer for seed production. And one strategy to improve nitrogen use efficiency in plants could be to direct nitrogen movement within the plant to where it's needed most. Now, generally in plants, nitrogen movement requires transport proteins. And our lab characterized specific transporters in the leaves of pea plants that when they were altered, it increased transport of nitrogen to developing fruits. And under high nitrogen fertilization, these plants produced more seeds. I hypothesized these plants were actually using nitrogen more efficiently and therefore would continue to outperform non-transgenic plants under reduced nitrogen fertilization. So I grew the transgenic plants shown on my slide in blue along with the non-transgenic plants shown in beige, and limiting deficient and sufficient nitrogen conditions. And under all conditions tested, the peas that had increased nitrogen transport to seeds were more efficient. Also, they had increased seed yields under all nitrogen conditions tested. And what's more impressive is when they were fertilized with half the amount of nitrogen, the transgenic peas still produced 14% more seeds than the non-transgenic plants under sufficient nitrogen conditions. My results showed that the transgenic peas not only were able to use the nitrogen within the plant more efficiently, but they were actually taking more nitrogen out of the soil supply. So overall, my work clearly shows that increasing nitrogen transport to developing fruits and seeds presents an effective strategy for crop breeders to potentially increase crop production while reducing the input of this nitrogen fertilizer. Thank you.